Welcome to Catch This. My name is Trudy Ray. You may have seen this story floating around in the news recently, because you will definitely look twice when you see a virus with an apparent vampiric stranglehold on another virus. This is a phage story, so don't go away. Satellite viruses lack one or more of the elements needed for replication, so they depend on co-infection with a helper virus that can provide the missing components. One well-known example of a satellite helper system is the hepatitis D, hepatitis B virus combination, where hepatitis D virus requires the presence of its helper hepatitis B virus to make copies of itself. But until recently, we were unaware of the existence of satellite viruses in the world of bacteriophages, viruses that infect bacteria. A new study has revealed two satellite helper systems in bacteriophages that infect Streptomyces bacteria. The phages were identified under the phages program, which allows students to sample soil and identify and name novel phages using a combination of microbiological and computational biology techniques. Phages isolated from the environment can be detected and quantified using a plaque assay, a technique that involves addition of purified samples, like from soil for example, to agar plates containing bacteria. If the bacteria on the plate are susceptible to infection with phages from a particular sample, the phages will infect and kill the bacterial cells and produce clear spots or plaques in those locations on the agar. The phages can then be isolated from the plaques, purified, and their genomes can be sequenced to determine their identity. The authors of the study noticed that purified isolates from soil samples thought to contain two newly identified phages called flare and mulch, each produced two different types of plaques, suggesting that flare and mulch actually each consisted of two different types of phages. Consistent with this observation, when they sequenced the DNA extracted from the plaques, they found two different sequences for each of flare and mulch isolates. A long sequence, presumably from a bigger phage, and a much shorter one, presumably from a smaller phage. However, when the phages isolated from the plaques were used to reinfect new bacteria, only the ones with the longer sequences produced plaques, suggesting that only the bigger phage from each of the flare and mulch isolates was infectious on its own. Based on these observations, the authors tentatively concluded that flare and mulch were two individual satellite helper systems. They also concluded that the smaller phages, the satellites, were dependent on the bigger phages, the helpers, for infection. The authors also noted that the short sequences were much more abundant than the long ones, suggesting that replication of satellite viruses using helper elements occurs at the expense of replication of the helpers. For ease of reference, they then named the helpers Mind Flare and Mulch Mansion, and their satellites were named Mini Flare and Mulch Room. The genomic material of phages is enclosed in a capsid head that is connected to a short collar and a long tail, which facilitates attachment and injection of genomic DNA into host bacterial cells. Phages are classified as virulent or temperate based on their replication cycles. Virulent phages actively replicate and lyse their host cell once their replication cycle is complete. Temperate phages either integrate their genomes into the host genome or linger in the host cell cytoplasm as circular double-stranded DNA plasmids until replication conditions become favorable. Satellite viruses typically infect a cell and stick around using temperate phage mechanisms until there is infection of the host cell by a helper virus. Now, the authors discovered that mulch room has genes associated with temperate phage properties, suggesting that it can remain in the host cell until mulch mansion infects the cell at a later time. But in contrast, Mini Flare does not encode any such genes, suggesting that it is likely a virulent phage that cannot integrate into the host genome or form a plasmid inside the host cell. This finding implies that Mini Flare needs Mind Flare for cell entry and must presumably wait outside of a prospective host cell until Mind Flare shows up. But this assumption raises the following question. How does mini-flayer synchronize its timing of infection with that of mind-flayer? I mean, it's a virus, so it doesn't have a brain. Perhaps the most intriguing discovery of the study was made by electron microscopy, with an abundance of images showing the tail of mini-flayer apparently wrapped around mind-flayer's neck. Although the purpose of this interaction is not clear, it is possible that the close association of the virus tails during attachment to a host cell helps both viral genomes get inside the host cell. If this is true, the exact mechanism by which this transfer occurs still needs to be figured out. Pretty cool story. Phage satellites make an interesting addition to the evolutionary tango between phages and their hosts. I look forward to learning more about how phages and other viruses interact with each other as imaging technologies and other molecular techniques improve. That's it for today. Thanks again for listening. My name is Trudy Ray, and I'll see you next time on Catch This.